Okay, so as we're waiting, we've been doing this traditionally now with all of our webinars, and that is to poll everybody who's on the call to see which databases you typically use from VEuPathDB resources. So you can use your phone and scan this QR code or type in this URL in your browser. I'll also put it in the, or one of us will put it in the chat channel so you can access it as well from there. And when you do this, let me go ahead and start this poll. And so as you start answering, we can see how many of you are, which database you use from VEuPathDB resources. So we'll let this go while we're waiting for other people to join. And I'll show my PowerPoint again in case you didn't get a chance to scan this or get the URL. I can copy this into the chat window. Also, the other thing to keep in mind as we're waiting, there is a handout that's associated with this webinar, and it's available in the handout section. So you should have a handout section in your GoToWebinar panel, and which will allow you to download a PDF, which will go along with this, some aspects of this webinar. So let me go back to here. We can keep looking. So people are still voting, so that's great. So we'll let this go for a few more seconds. Looks like there's a race between FungiDB and TriTripDB in terms of most number of people, at least people who are voting. And I'm still seeing the number of attendees are increasing, so I'm gonna give people maybe a 20 more seconds before we start the webinar. So again, for those of you who are just joining, feel free to scan this QR code or go to this website. And this will allow you to vote and let us know which resource you use from VEuPathDB resources. We've been running this poll in each of our webinars, and so it's been interesting to see what databases people use. All right, I think this is a good number of people who voted from the attendees. So we'll go ahead and get started. I'll keep this poll open, so feel free to vote anytime, obviously. And if we have time, I'll show this again towards the end of the webinar with the link, so you'll get a chance to vote again on your on the databases that you use most frequently from VEuPathDB resources. Okay, so welcome to this week's webinar. So as many of you know, we've been running weekly webinars for the past 2 months, about 2 months. All of these webinars are posted, we advertise them in different places. We have a page here that where we post all the webinars, and I'll show you what this page looks like. So this is the webinar page. We have upcoming webinars and previous webinars. All the previous webinars are linked to the YouTube recordings of this webinar, which is a reminder that this webinar is being recorded. So if you miss something, you'll have an opportunity to go back and visit or review that webinar. We also have links to the handouts. And of course, we encourage you to share these with your colleagues. And so we understand that not everybody can attend these webinars at the time we have them scheduled. So feel free to share them with folks in your labs or colleagues of yours in your department who you think may benefit from these webinars. And just one housekeeping or a couple of housekeeping things about GoToWebinar. So during the webinar, if you have questions, feel free to use the question panel in your GoToWebinar control panel. So just type the question in there. And we have a number of people from the VEuPathDB side who will be helping in answering questions. These include Gloria Geraldo Calderon, who's based in Colombia, Sam Rund, who's based at the University of Notre Dame, and Suzanne Warrenfels, who's based at the University of Georgia. And today's webinar will be mainly presented by myself, Omar Harb, and Evelina Beshenko, or Eve Beshenko, who's based at the University of Liverpool. And in case you didn't know, today's webinar will focus on Go and other enrichment analyses that you can conduct in VEuPathDB. So before we get started, our outline for today's webinar is first going to focus on, well, what is a gene ontology? The other thing we're going to look at are what are enzyme commission numbers and how they relate to metabolic pathways. Where does VEuPathDB get Go Annotation EC numbers? And I'll just mention this, it's nothing magical. What is an enrichment analysis? Again, very basic terms. And then we will run through a number of demos to highlight how you can actually take advantage of these types of searches in VEuPathDB resources. So before we delve into Go or gene ontology, the question is, well, what is an ontology? And in the case of the way scientists use ontology, it's really a description of what exists in a particular field and the relationships between them. And so you can put together an ontology, a very basic one, by simply describing the field of science. And so this is something I just put together last night and trying to see how I can explain this, hopefully in an okay manner. But the idea is that you can take something like the field of science. We all know what science is. It's divided up into three general subcategories: formal sciences, natural sciences, and social sciences. And then you can describe each of these terms here, in a standard way that everybody can understand. And then each of these has subcategories and so forth. So these will go on forever, but also there are relationships between these different categories. So you could say, well, mathematics belongs to formal sciences, which is a science, right? And you may reach a point where you can say, well, in physical sciences, that includes chemistry, right? And a lot of chemistry could then be linked to biological sciences in terms of describing or explaining some of the biological functions that you are working on in another field or another realm of natural sciences. But the idea behind it is that you have a formal description of the knowledge within a particular field, and importantly, a hierarchy of that knowledge. So if something belongs to something else, or something is related to something else in a certain way, right? And so these formal descriptions then allow you to use this information for useful things downstream, like Go enrichment analysis, which we will talk about today. And so when it comes to gene ontology, right, we can, gene ontology was developed in the field, again, we're part of the life and biological sciences. Gene ontology was developed really to describe the knowledge available in the biological sciences, and also the relationships between all this knowledge. And the way the Gene Ontology Consortium did this is by dividing this knowledge into three broad categories, and those are molecular function, cellular component, and biological process. Each one of these serves a particular purpose. So, and here's just a quick description. All of this is coming, so I provided links in my slides, which are also in the handout. There's an appendix, and there are links there as well. So you can read a lot more, obviously, from the Gene Ontology website. And most of these descriptions are obviously coming from that website. So molecular function really describes the activities of the gene products, right? So a toxin activity, for example, or a catalytic activity, or a transporter activity. So these are all are things that describe molecular function. And then each one of these will have subcategories and potentially more subcategories, right? And so an enzyme, a hydrolase, for example, could have some kind of catalytic activity, right? And so you can imagine building those relationships. Cellular component describes where a function takes place. So the subcellular localization, or the cellular location of where that function takes place. So like the mitochondrion, or a plastid, for example, or cilium. So those are all cellular components. And again, each one of these comes with a detailed description links to synonyms of the term. For example, the word psyllium is also often referred to as flagellum, and other, depending on your domain. And so these are all things that a Go ontology helps with. Describing and making sure that you're talking about the same component or category when you're discussing function or cellular component or biological process. And the biological process really describes the process accomplished by multiple activities. So like some kind of a biochemical pathway, for example, like pyrimidine biosynthesis. So why is this important? So why should we do this? And so I'm going to just provide an example of a basic biological example. So these are all different organelles found in different organisms. So cyanelles, chloroplasts, and an apicoplast. And people here who work on Plasmodium or Toxoplasma know what this organelle is. But these are all plastids, right? They all belong to this category plastid, right? And so knowing this, knowing that all of these are linked together under plastid, then provides you with an ability to start asking questions and finding relationships between functions that occur in these organelles and functions, functions that occur between these organelles, right? And they're linked by the term plastid, right? And so that then allows you to really expand your knowledge and discover things across the evolutionary tree, basically, by using Go terms that link them together. And so where do Go terms come from? And so they're really two basic places. So either you have an annotator, somebody who looks at a gene and says, oh, this gene looks like a hydrolase, and so I'm going to give it this Go term, and it is also belongs to has this type of catalytic activity, so I'm going to give it that Go term. And you then place it, you give it a particular ID, like in what's shown here as a plastid, it has a Go term ID. And so if your particular enzyme, like ferredoxin NADPH reductase, for example, let's say its activity takes place in the apicoplast, you would give it the Go term for apicoplast, but also that enzyme's function will fall under the category of plastid as well, okay? And so you can manually do this, right, as an annotator. You can transfer it by similarity, right? So you can look at other organisms and say, well, the homologue of my gene in plants is in the chloroplast, so I'm going to give it the plastid Go term. But again, it's still a manual effort. You can do it automatically, right? You can say, well, just transfer it. As long as it's similar to a certain extent, I'm just going to transfer that Go term, okay? So that becomes more automated. And then finally, you can transfer them based on domain similarity. So if I know that my enzyme has a reductase domain, for example, in it, then that would, I could transfer in a particular Go term that's associated with reductase domains, right? And so, and there's a standard way of doing this called interpro to Go. So typically people can run something called interpro scan on their genomes, which defines various types of functional domains in their genes. And then you can transfer basically or link between an interpro domain and a particular Go term that's associated that describes that interpro domain function or biological process or a cellular component, right? And so that's a very nice way to also automatically transfer Go terms to your gene of interest or genes of interest. So I'm going to switch gears a little bit and talk about enzyme commission numbers. And so this is basically a systematic and logical way to describe enzymes, right? And you've probably seen these before in metabolic pathways. So they usually look like a 4-digit number separated by periods. Each one of these digits has a particular significance. And again, there's a link here, which is also in the handout, where you can read a lot about all these different structured way of describing enzymes. And so you could see here that the first number shows which of the 6 main divisions or classes the enzyme belongs to. So they divided enzymes into 6 main classes. And so you can, for example, hydrolase is one of those classes. And so you can, if your enzyme is a hydrolase, then you'll give it the number 3, right? You may know more things about your enzyme, so you can give it additional numbers. So if you know your enzyme for example, you know, its subclass of this hydrolase, if you know, for example, it acts on ester bonds, right? Then you would give it the number 1. So 3.1, you will immediately know that this enzyme is a hydrolase that acts on ester bonds. Now, if you even knew more about this enzyme, right, about its function, and you knew, for example, that the subcategory of this ester bond that acts on ester bonds, but that it's also acts on phosphoric ester bonds, right? So then you can give it another number here, so like 3.1.3. So EC numbers can be complete. They can include the 4 digits, like in this case for acetylheptulose 1,7-bisphosphatase, it's 3.1.3.37, or they can be incomplete. For example, if I only know that my enzyme is a hydrolase, it may have a number like 3. Right? So I don't know anything else about its subclasses. And that's important as you investigate metabolic pathways. You always have to ask, well, do I really know that this enzyme is specifically acetylheptulose bisphosphatase, or do I just know that it's a hydrolase that acts on ester bonds and it's phosphoric in nature? So it could be acetylheptulose, but it may also be a fructose bisphosphatase, right? And so those are all things that that would come into play. But obviously, a complete EC number with the 4 digits gives you down to the specific enzyme and its function, basically. And where these numbers come from, again, by manual annotation, that's one of the main sources. But also, you can transfer these by similarity. So you can say, well, if my gene is at least whatever 90% similar to another gene that has already an EC number associated with it, I'm going to just transfer that EC number. So that's very useful when you're trying to do automated whole genome transferring of EC numbers. But of course, this is error prone, right? There will be cases where there are EC numbers that are wrongly assigned and that wrong association will get transferred. So again, as a biologist, you always have to look at the results you're getting and determine whether you trust them or not. But these EC numbers then can be used. This is just a little snippet of a metabolic pathway from VEuPathDB site. So we show KEGG and Metapsych pathways all displayed in Cytoscape. But the point here is that you'll see these different compounds, so products and reactants in a biochemical pathway. And they're linked by enzymes. And these enzymes are denoted by these EC numbers. So 1.2.4.1, that's an EC number, and that represents an enzyme, a specific enzyme activity that is present and associated with a specific gene or a specific subset of genes in the genome. So if your genome if you're able to annotate your genome with EC numbers, you can then transfer this to metabolic pathways and populate metabolic pathways with these EC numbers, and you can start asking, well, does the Calvin cycle, does it exist in my particular organism, right? Do I have all the enzyme components present in there, or is it only one enzyme out of 10 that should be in the pathway? So maybe that pathway doesn't really exist. It's just that particular reaction that exists in my organism of interest. So both EC numbers and Go terms, or any way you can describe functions of genes, can be used in enrichment analysis. And this is really useful, basically, if you have a list of genes that came out from your, let's say, your proteomics experiment or an RNA sequence experiment or a microarray experiment, and you have, let's say, a list of 200 or 300 genes that are the most interesting one by whatever criteria, and you're wondering, well, is there anything interesting here? And is this interesting thing that I'm seeing in my list, is that just by chance that they came up in my list of upregulated genes, for example, or are they truly significant? Did they, am I getting a true observation here that may have obvious ramifications downstream? And so typically when people want to, and this is an enrichment analysis, you're asking, do I have anything enriched in my gene set of interest compared to my genome, the background, right? There's a really nice, and typically people use something called the Fisher's exact test. There are many different types of statistical tests that you can use to determine enrichment. Fisher's exact test is one that is frequently used, in particular when you're doing Go enrichment. It's typically used for contingency tables, and in fact, it's actually typically used for small sample sizes. And this poses a problem, right? Because in the case of a genome and the types of data we're looking at, your sample size is actually quite big. It's bigger than what Fisher originally designed his statistical test for, and so this poses some problems because it affects your p-value. So Fisher's exact test will generate a p-value for you. And as we know, we typically use 0.05 as a standard for, if it's above 0.05, then this is not significant. If it's below, then it is significant. So this means that there's always, for any test you do, there's a 5% chance that that you got this by chance, right? But when you have hundreds or thousands of tests, let's say in the case of a genome, you're going to every single Go term can be considered as a separate test, right? You're testing whether it's present in your sample by chance or not, right? Then each one of these tests will have a potential 5% randomness possibility, right? And so this will add up, of course. So your p-values then from a Fisher's exact test will become inaccurate. And so that's why then typically people do some corrections to this multiple testing artifacts. And typically they run something called either a Benjamini-Hochberg false discovery rate or a Bonferroni adjusted p-value. And you will see these. So this is just a screenshot, and Eve, who will be presenting next, she will run through a number of examples in our various databases. And what she will be doing is showing the results that look like this. And she will describe most of these columns. All of these columns have question marks next to them, which you can mouse over and get more information about the different things. But for example, you have the genes in the background with a particular Go term. How many in your sample set had that Go term? Is this, you can see what fold enrichment this is in your sample compared to the background. Fisher's exact test generates an odds ratio as well. And this really, typically an odds ratio above 1 is considered, good, and below 1, it's at least means that there's no association. I'm not 100% sure how useful it is in our Go enrichment analyses, but nevertheless, it's an output that we provide. The p-value is also a product of the Fisher's exact test. And again, for each of these, for example, 18, there's a probability of it appearing, not by chance or probability of it appearing in your sample. And then for all 18, each one of them has a probability. And so this is an overall p-value for what are the chances of all of these appearing in your sample. And you'll see here that this is a pretty good p-value in this case. So you would say, well, this is significant. And but as I mentioned, since this is a large samples that we're talking about, we do apply these corrections. And you'll see that the corrections always have a lower p-value, right, or a less significant p-value. And for things that are very significant, in most cases, they don't necessarily, it doesn't really make a difference whether it's, 10 to the minus 9 or 10 to the minus 11. Both of these are significant, but it becomes more important when you're looking at things that are less significant. So when you're getting to things that maybe, at the 0.05 significance p-value for the Fisher's exact test, but with Bonferroni, it goes above that, and so it's less significant. And so those are the things at the edge cases that then it becomes really important to look at the corrections for those enrichments or for those p-values. Okay, so I'm going to stop here and switch you over to Eve, who's going to demo some of these. And as she's getting set up, I'm just going to mention that we've been as I'm sure many of you know, we've been working very hard on putting together a beta sites for all of our databases. This is, as many of you know, this has to do with our new funding from the Bioinformatic Resource Centers at NIH, and it constitutes a merger between VEuPathDB resources and Vectorbase. And the cool thing about this is that these beta sites are built under the same infrastructure, so all the tools are available to everybody, vectors and parasite biologists or fungal biologists, and so that's pretty cool. And these beta sites are actually out now. They've just been released this morning. We'll make some announcements about these very soon. But I will, after Eve is done, I will show you Go enrichments in the beta site. In particular, I demoed this in vector-based. So this should be pretty cool for those of you who work on vectors. But the same functionality obviously applies to all of our resources. All right, Eve, I'm going to hand it over to you.
All right, thank you, Amar. All right, I am on the FungiDB. I'm on the FungiDB website, and I'll show you a few searches that you can do within this website. And similarly, they work on essentially all other VEuPathDB sites as well. So I have pre-run a search strategy that essentially looked at the transcriptomic dataset in Cryptococcus neoformans, and then subsequently identified how many of these genes also have a predicted signal peptide. But for our purposes today, we'll take a look at the genes that I have found in step 1. So the significance of this search is actually, I'll go ahead and show you the revision screen so that to show you the samples that I have used. This is a sample where Cryptococcus neoformans cells, a fungal pathogen that causes human meningitis and invasive diseases in humans. The cells were grown in yeast extract media known as YPD, and that is also a control sample. And they were also, the dataset also had several samples such as in vivo. So what I wanted to see is actually take the same strain grown in the control media and in cerebrospinal fluid and determine how many genes were upregulated in the in vivo stage of the experiment, and to see if we perhaps could find something cool when it comes to enriching for the function of gene. So I'll go ahead and close this window. To deploy Go Enrichment Analysis, you essentially will be working with the gene results table that summarizes all of the genes returned in your search. And for my search, I have identified 168 genes that have 4-fold or more upregulation in their expression in the in vivo stage. And the next step, I would like to essentially perform a Go enrichment analysis on these results. To deploy the tool, navigate to the Analyze Results tab, which is shown right here in blue. And once you click on the tab, the screen will refresh and essentially will ask you what is it that you would like to do with the results that you have just generated. There's 3 options, and so essentially all of the 3 types of analysis apply: Fisher's exact test to evaluate ontology terms or overrepresented pathways if you're working with metabolic pathway enrichment tool, or product description terms that occur in your results. And essentially, when you navigate to the gene ontology enrichment, the screen provides additional parameters that you can use to set up your search. So as Amar mentioned before, Go term data can be mined within the 3 sections, so to speak: the functional description of genes that's corresponding to their biological process, cellular component, or molecular function. We also provide an option to select whether you want to look at the computed or curated information, and normally the default parameter is set to select both. So computed simply means that these Go annotations were produced as a part of the electronic that were inferred from electronic annotation, and as such denoted as computed, while others have some degree of curation. And the default p-value cutoff is set up to 0.05, but you can obviously change that if you prefer. There's also one option that allows you to limit your search to GoSLIN terms. And so in my search, I will go ahead and select no. I would like to essentially run Go enrichment on all Go terms. But if you select GoSLIN terms, it essentially provides you a summary or brief overlook of the major terms enriched in your sample. So for example, Go terms usually refer to the original full set of annotations that are maintained by Go Consortium. And Go terms are granular terms that provide information about biological function or other processes. And here's an example of a tree that you will frequently see for individual Go components. Notice that it responds to drug or food or nutrients or other environmental stimuli are within the response to stimulus section. And that's essentially all of these Go terms are granular components over larger components that is defines them as a response to stimulus. So when we talk about GoSlim or running Go enrichment using GoSlim, you're essentially using the cut down version or a slimmed version of Go ontologies that containing only a subset of the terms to describing essentially all of the subfamilies that are included in the response to stimulus family. And that's there's no really strict rules of when you should use Go or GoSlim analysis, but you may imagine that if you have a large scale, high throughput data sets with thousands of genes, you're not really maybe interested in knowing how many of genes respond to tagged with a response to drug or food. You maybe just want to look at how many of these genes respond to various type of stimuli and how is this different from, let's say, another high throughput data set that you have run using different conditions. And that's where GoSlim comes in. And it's quite handy sort of to give you a glimpse on the overall trends that occur within your data. Okay, so in our experiment, we will go ahead and leave all the parameters at default, click submit, and notice that I have already pre-run this search previously. So we'll just navigate to the results that I have. We'll just refresh it. And as with any analysis that you run in FunGDB or other VEuPathDB sites, the analysis will generate essentially a table that has a list of Go IDs because we're running a Go term enrichment analysis. Each of the Go IDs are described as to what term is being associated with this particular ID. And you also get a few numbers that Amar already have gone through, but the ones that we're looking at, for example, genes in the background, that means that there are 333 genes associated with this particular Go ID, such as transmembrane transport that are annotated in Cryptococcus neoformans H99 in Go Ontology, and 16 out of your genes are actually matching to the data set that we have looked right here, those 168 genes. Okay, so there's various things that you can do. For example, you can use the either p-value or Bonferroni p-value to sort your data and bring up, let's say, those hits that have essentially the higher confidence levels, and to see how that changes, and also what are sort of the top hits that you get. And for us, whether you sort by p-value or by other p-values, it really hasn't changed the output. And the Bonferroni, I just wanted to mention, the Bonferroni essentially is a method that corrects for multiple comparisons when it comes to p-values. As far as the further analysis of your results, you can always export the data and save data on your computer if you would like to go back and look at the results. But you can also further analyze your results, such as visualizing it. We offer a way to sort of graphically see your data if you click on the Open in ReviGo button, and that essentially redirects you to a site that will take the Go terms and associated p-values and help you to visualize your data. So ReviGo is essentially a web server that takes, or rather summarizes, the unintelligible lists of Go terms and associated p-values, and then essentially finds a representative subset that it can cluster all of these Go terms under. It's essentially an algorithm that relies on semantic similarity. So let's go ahead and run the search. And I will open the tab that I just pre-ran a few minutes ago. And that's one of the graphical outputs that you would normally see associated with Go Enrichment Analysis. There are a lot of publications available that describing how the algorithm works and giving you a little bit more information of what things are. So I recommend that you find those publications, and I think one of them is also linked in the handout section. But so what you are viewing right now, it's a scatter plot and table where all Go terms and their associated p-values have been mapped in essentially 2D space. And there's a few things to take a look. For example, the color indicates the user provided p-values, those p-values that we have imported from our analysis. And the blue and green indicate more significant p-values compared to the orange and red. And also the size of the circles indicates the frequency of the Go term in the underlying Go database. So you would expect the larger circles having a high frequency. And in addition, the proximity of the dots themselves reflect the semantic similarity. For example, you may expect that if you scroll down and see what terms are actually mapped here, all of this is related to transport. And so far, every single dot in one way, in one or another way, has been related to the transport function. So there's one more thing that I wanted to mention before I actually showed this screen. One is really cool thing about Go that it also essentially reduces functional redundancy. And what that means is that it takes multiple Go terms and looks whether it can find an overarching Go term that is essentially including all of the sub-Go terms, and displays this underneath the graphical visualization. For example, all of these three terms have now been reduced into one main that is associated with this Go ID. And you can choose to hide and show only the Go terms that are left after the reduction, or you could choose to expand to view everything, essentially all components that have gone into build these graphs. And there is additional information available for how to use various maps on the ReviGo site. Okay, so going back to our strategies, one thing I want to show you really quickly before we move on to looking at metabolic pathway enrichment is actually that you can, in addition to viewing the Go term enrichment, you can also build word cloud maps. And these are available essentially from the Go enrichment page and located above the table results. If you click on the show word cloud, it essentially will take in the Go terms produced by your search, associated p-values, and will build a word cloud that you can download and potentially use it in your presentation if you choose so. But looking at the p-values, notice that the transmembrane domain, carbohydrate metabolic processes, and pathogenesis are one of the higher hits and correspond to the word cloud shown. All right, so let's switch gears and talk about metabolic pathway enrichment. I would like to show you a strategy that utilizes essentially a number of data sets from Phenotypic Evidence section that we have. So I'll navigate back to home, and the data can be accessed underneath the Phenotype Evidence within the Search for Genes bubble. So if you click on Phenotype Evidence, we have quite a few data sets integrated for various organisms, including data sets integrated from various providers, manually created data by VEuPathDB, and provided by individual labs. So I will search the data set from Firebase. And to search the data set or deploy the search, just click on CP, which stands for Curated Phenotypes. And on the bottom, a new window will expand, essentially offering all different types of metadata that can be searched using the menu on the left. For my experiment, I am interested in seeing anything that relates to aspergillosis. So let's take a look at invasive, essentially, aspergillosis, and include pulmonary aspergillosis as well, and click on Get Answer. So what the search is actually doing is searching the records that are associated with any disease phenotype caused or associated with Aspergillus species. And if you scroll down and actually look at the results, you have 157 genes. And furthermore, you notice that we have a lot of fumigators genes, etc. But if you look within the Aspergillus species, you notice that there are several Aspergillus species are returned within our hit, meaning that the records that we have and provided by the 5As essentially have a disease phenotype curated to this particular species and specific genes associated with it. So one way you can further enrich these results, and one of the interesting things for me would be to look at, for example, metabolic pathways that may be enriched when cells are growing in human tissues or undergoing an active process of invasion and dissemination. So one way to perform a metabolic pathway analysis, again, click on the Analyze My Experiment tab, and then on the Metabolic Pathway Enrichment tool. So the interface is quite similar to what you have seen in Go Enrichment. Notice that if we click on the organism drop-down menu, now we have multiple organisms available, and for statistical reasons, you can only run one search at a time. So let's go ahead and look at Aspergillus fumigatus, AF293. And I will leave my criteria at default, which means we want to look at the CAG and Metasig databases and see what we can find, what type of pathways are enriched in our results. I'll navigate to the tab where I have already pre-run this experiment, and essentially this is my my results. There are several pathways that seem to be enriched in those cells that, or rather in genes that have been annotated with a human pathogenic process, such as invasive or pulmonary aspergillosis. And interestingly enough, there's also steroid biosynthesis and ergol biosynthesis that we know play important role when patients undergoing antifungal treatment, such as we know that some of the isolates from Aspergillus species have multiple resistance to antifungal drugs due to mutations in the ergosterol biosynthesis. So this is quite interesting, and if you were to look further and see potentially maybe some of those genes actually have SNPs that may be of interest, you could run additional searches in FineGTB by essentially adding STEP to your current strategy and intersecting results with other data sets. For now, let's take a look at ergosterol biosynthesis 1 pathway. And you can do this essentially by clicking on the link described on the link here. I already have the pathway open in the tab, and this is quite a long pathway, but what I wanted to show you is that the pathway can be explored in the Cytoscape.js window. It's an interactive display to where you can drag and drop and reposition some of the nodes annotated within the pathway. And this particular display is also powerful because you can display the results of various data sets by overlaying them on the nodes. For example, in this particular example, I have already generated an overlay by painting enzymes by experiment. Because we know that ergosterol pathway is important in drug resistance, I have chosen to overlay the experiment in a fumigatus cells that have known drug-resistant phenotypes. So to paint your various experiments onto the node, just navigate to the paint enzymes by experiment, and then to filter results for fumigatus specifically, I just typed in the first few words of the fumigatus at the top and essentially activated an RNA-seq dataset that is integrated in FungiDB, and that provides expression values for various strains that have different types of antifungal drug resistance. And essentially by painting this over, not only that you can click on individuals' notes and to see EC numbers, specific pathways, specific enzyme names, but you can also see the visual representation of the transcriptomic data associated with each component. And for some, you may be able to see that there's a general trend across all isolates, for example, to increase the expression of certain nodes in response to drug, while in some you will have a differential expression. Okay, I think
That's
this
great.
is
So
all
Eve,
for my presentation.
Great, thank you, Eve. So there are a number of questions that are coming up that I wanted to make sure to address. I'll probably just address them verbally. It'll be easier. So there was one question, or you can address them, Eve. I didn't mean I should address them, but there was one question: is it possible to perform a Go analysis with a new set of a new data set? And so, for example, something that's not in our databases, but and I think. You want to go ahead and answer that, or you want me to go ahead?
Yes, I'm assuming a new data set meaning a custom set of genes or a list of genes. Mm-hmm. Yes. All right. Yes, you absolutely can. Obviously, you have to be aware of the fact that the quality of Go annotation and the availability of the terms is different for all genomes. Some genomes are very well studied and have been manually annotated, while others annotation may be produced by inference using electronic annotation, and as such you have to be aware of the caveats that exist. But if you have a list of genes for the genome that is integrated in FungiDB, you absolutely can do this. First, it will be to essentially upload the list of genes into FungiDB, and you can do this by navigating to the annotation, curation, and identifiers, selecting gene IDs, and essentially if you have an Excel spreadsheet or a text document, copy and paste gene IDs into a window that I will show in a few seconds. Here we are. Essentially, gene IDs can be pasted here. Once you click on Get Answer, the IDs will be taken up, and what will be returned is essentially a search strategy that looks like this, with the first step being a list of the custom gene IDs that you have.
Great, thanks. So another question which we answered, but I thought it would be good to bring up, and I can go ahead and answer it because I answered it by when I replied to it in the questions. But the question was, what about hypothetical genes? How do they relate to Go terms? And that's a really good question, because often in genomes you have, hundreds of genes that are annotated as hypothetical, so they don't have a product description. So the question is, well, how do these, how can you use these in Go enrichment? And so the answer really depends. And so for many of those hypothetical genes, we actually can associate a Go term with them, because a hypothetical gene, even though it doesn't have a product description, it may actually contain functional domains. So when we run interproscan on the genomes, or typically people who do gene annotation, they run a scan for PFAM domains or types of functional domains in a genome. And so you can have a hypothetical protein that has a kinesin domain, for example, and that domain then can be linked to a Go term. In this case, it may be linked to motility, because the kinesin domain may be in a protein that binds to microtubules, for example. And so So that's how you can imagine having a subset of hypothetical proteins that will have domains, functional domains, and will, as such, will have association with Go terms. There will be obviously cases where it's a hypothetical protein with no information about its function, and of course, then it won't have an associated Go term. So,
yes, that's correct.
Mm-hmm.
And if I just may add a few things, I have added a step to a strategy that I have shown, essentially looking for hypothetical in the gene product. And surprisingly enough, you have 96 genes out of 168 in Cryptococcus neoformans 899 that do not have a essentially well-defined product. That doesn't mean that they will not have Go annotations. A higher overview annotation, such as the ones that I have shown you before, response to stimuli may be annotated, or the genome may have been run through UniProt, and we may have some information, but you won't really know until you actually see what type of information is associated with this type of genes. And you could run Go Slim, or you could run both actually and see how the results are different. And for those genes that obviously have no annotation, no Go annotations, you will be probably a good procedure will be to look at all supporting data, including orthology and syntheny profiles from other organisms.
So Eve, in the interest of time, I'm going to take over as presenter, and because I'd like to show this functionality in the new beta sites, and this is in particular useful for the vector-based community who will be getting access to these tools, at least in the VEuPathDB world, for the first time. So go ahead and show my screen. Okay, so as I mentioned, at the end of my section at the beginning of this webinar, we've been working on generating or developing these new websites, which integrate all resources in VEuPathDB. And I will ask, other outreach team members who are on the call to go ahead and paste the URLs, so to the beta VEuPathDB site. But in particular, you can go to beta.vectorbase.org, and you will have access to pretty much all the data that's available in the legacy vector base, which is still obviously an available resource for the time being. But there will be many tools that will become available to you through this beta resource. And so, for example, the analysis enrichment or the enrichment analyses tools that we talked about today are available to you if you are a vector-based user or any of our database users. So what I would like to do is quickly demo how you would do this in the new beta sites. Also, if I can have maybe Suzanne, I think you're on the call, maybe you could go ahead and also post the link to our scheduled webinar introducing the beta sites, which is in a couple of weeks. And this will also, for others who are interested in getting a general overview of these sites, this will be a really nice webinar for you to join. Sure. So thank you. So what I'm going to do is I'm just going to quickly, in this new vector-based beta site, I'm just going to scroll down. I'm going to run a quick transcriptomic search. I'm going to pick a microarray evidence search just to do something different. And there are many, many experiments listed here. Obviously, you can walk through them one by one to see which experiment you may be interested in. In my case, I'm interested in a Plasmodium infection experiment. So I'm interested in finding genes that are upregulated in cells, mosquito cells, that are exposed to Plasmodium. And so there are many experiments here that potentially fit the bill. And I'm going to pick this one here at the bottom, which is used hematocytes from Anopheles gambiae. So we can go to the search. So this opens up the search page here. And I'm looking for genes from this experiment that are upregulated. I'm going to keep this at twofold. And in this particular experiment, they compared an infectious parasite to a non-infectious parasite. And so I'm going to compare the infectious ones to the invasion deficient ones. And I'm looking for genes that are upregulated in my infectious ones compared to my invasion deficient ones. So in theory, things that are potentially important from the mosquito side during the infection process. So I'll go ahead and click on Get Answer. And so as Eve showed you before in the legacy sites, it's the same idea. It's just built on a new infrastructure, modern web architecture. And so you get a step in a strategy here, which returns 178 anopheles genes that were upregulated by at least twofold in in this particular experiment. You can scroll down and look at these genes, and you'll have a graph here that represents this expression in the two samples. So you can confirm that these are the right types of profiles that you're interested in. And then you can start, either analyzing these and saying, well, I want to see if there's anything enriched in these, or you can further narrow them down. And so as Eve showed you, you can add steps. So I'm going to add a step. Again, it's the same mechanism, just it's a bit more modern and also built on lots of years of experience. So we've improved this interface. So I'm quickly just adding a step. I'm going to intersect my results from my previous step. And I'm going to ask, well, how many of these genes that are upregulated during invasion in the mosquito are also potentially secreted? So I'm going to look for any gene that has a predicted secretory signal peptide. I'm going to select all organisms in VectorBase, and I'm going to run this. So now I get a two-step strategy. So out of these 178 genes that are upregulated during invasion are also potentially secreted. And so here's my list of genes. I can quickly get a word cloud based on the words that are in the descriptions, right? And as you see this, you will see that there are many words here that are not really terribly useful for defining function. So I can filter these out. So I can immediately see, oh, there's the word appendage appears 6 times if you mouse over it. And, the word sensory appears also 5 times, and so forth. Serpent appears 4 times, right? The question is, however, is this really enriched? Is this just happening by random, or do I have an enrichment here? And so this is where obviously the enrichment analyses we were showing you are very useful. So quickly, as I scroll down here from my gene results, I can look at the genome view if I want. And so this shows me the distribution of the genes across the different chromosomes. And so I can see that 2R has 35 genes on it, and here's the distribution. So this is a nice and quick and dirty way for you to see the distribution of the genes on the chromosomes. I can click on Analyze Results, as Eve showed you. So I'm going to quickly click on Go Enrichment. And then as this appears, I'll just pick, let's say, molecular function and submit this. And so this will run and give me a list of results that are potentially enriched. And as you can see here, there are a bunch of serine type endopeptidases, various peptidase activity that are highly enriched or with a really good p-values. And this is exactly, in fact, it turns out, I looked at this before the webinar just to confirm this. It turns out the paper specifically highlighted these types of functions as being important and things that came up in their experiment. So that's quite nice. I want to point out one more thing. So Eve indicated this metabolic pathway. Some of you may have noticed that we have this word enrichment. And so what this word enrichment does, it takes the words that are in the names, the product descriptions, and does an enrichment on those, right? And the advantage here is that you can imagine having gene families in a particular organism which do not have a Go term. There's just no Go term for them, but it's a gene family. So imagine some kind of a variable gene that's expanded in a particular organism, and those will not come up in a Go enrichment analysis because there isn't an associated Go term. Or even the word hypothetical, right? You can imagine enriching for hypothetical. You can say, well, my hypothetical genes are enriched in my sample. So I can go ahead and run actually an enrichment analysis on the names of the products, right? And you'll see here that the word serine, for example, is highly enriched in my experiment, right? So they're most likely serine proteases, clip domain proteases, right? This appeared, it didn't show up as there, but a clip domain is a protease as well. So technically you got this in the Go term enrichment, but doing this on the words also gives you something about the names and the product descriptions and how they are enriched in your sample set. So that's another sort of companion enrichment analysis that's useful to do. So I'm gonna stop here, and of course, people feel free to continue asking questions. We'll stay on for a few more minutes and see what other questions come up. And others on the call, let me know if you see any questions that need answering here. I'm looking at the questions as well. And again, as I noted, these beta sites just came out this morning, so this is really a nice early preview that you're getting. We'll be making a bunch of announcements over the next few days, sending out emails and social media and so forth. So you will probably get inundated with information about the beta sites, but we are really looking for your input. So you as users of our resources are really influential in driving what our resources provide and how we, and guide us in the next few years. And so in fact, we do have a number of surveys available. If you go to the news section of the beta sites, of any of the beta sites, you will see that we have a few surveys available that we encourage all of you to go in and try them out. This will do two things. One, it'll give you some training on the new resource, but at the same time, it'll give you an opportunity to let us know if we got things right, if things are working properly, if things can be improved, and any other suggestions you may have. So these are quite useful, and you'll be hearing a lot more about these, but I certainly encourage all of you on this call to try these out and give us your input. Okay, I'm trying to see if there are any other questions. So Eve or Suzanne or others, are there any questions that we should look at here, or are we good?
Looks like we answered them.
Yep.
Yeah, so all beta sites, so there's a question quickly, are all beta sites available for all sites? And the answer is yes. So you can go to beta.fungidb and you'll get access to the beta site in FungiDB. You can go to beta.plasmodb.org and you'll have the PlasmoDB beta site. You can go to beta.trickdb.org and you'll have the Trichomonas database in the beta version. They will all look the same, and the same functionality will exist on all of the sites. And so that's one of the nice advantages of building all our sites on the same infrastructure.
There was also a test about how this relates to COVID-19 and how this study can be used to understand COVID.
Yeah. Yeah. Did you want to answer that, Suzanne, or should I go ahead, or
do you
have an answer?
Go ahead.
So the question was, how can this study be used to understand COVID-19 and its treatment? So in VEuPathDB databases, the genomic resources at least, we do not provide access to COVID-19, but we have a sister bioinformatic resource center. So the virus bioinformatic resource center, which provides access to COVID-19 genomes and may be worth investigating there. In terms of enrichment analysis in general, those can be used anywhere, right? So in any case where you have a set of functions or names that you want to determine whether they are enriched in your sample compared to a background, you can use an enrichment analysis, right? There's an example that we provided a link to in the handout, or Eve provided a link to in the handout, to a YouTube tutorial describing how you can use a Fisher's exact test for enrichment analysis. And the example that's used, which is quite nice, is using a bag of M&Ms, right, which is chocolate candies which have different colors. And in a bag has, let's say, 10 blue ones, 5 red ones, 6 green ones, and so forth. And you're asking, well, what are the chances that I'm going to get 4 blue ones and 1 yellow one in that order, right? And so you can use a Fisher's exact test to do that. And so clearly you can use an enrichment analysis for any of those types of analyses. Yeah, and so there's another question about Glossinidae. Is there any information like this in VEuPathDB? And so I'm just going to go back here to VectorBase. And so I'm going to use the beta site just because it's available to you. And so you can go in here and easily look at, you can look at what genomes are available in the database in different ways. But one way is you can go under organisms and you can go under genome data types, let's say. And this will show you that there are 40 organisms in here, and you can go down and look at all the organisms that are available. And so you can see that there are a few Glossina species available. And what's available for each of the organisms obviously depends on what data is available. So we load data sets for the organisms. You can be very influential in this as well. So the way we hear about these data sets is by attending meetings and so forth, but also by getting emails from you letting us know about data sets that are important. And of course, we scour the literature on a regular basis to identify new and important data sets. And this webinar is recorded, as I mentioned before, and you will get a link to the recording, and we'll post it on the webinar page as well. So there's a question about what is the difference between, let me see if there's I think we've answered all the questions so far. Oh, somebody's asking whether we get an e-certificate for the attendees. So GoToWebinar has a function of providing an e-certificate. We're happy to generate one. We typically do not do this for the webinars, but if you need one, we're more than happy to do that. As usual with all of our websites, we have a contact us link. So feel free to contact us anytime with questions, suggestions, or issues you may have with the website. And of course, if you're one of the people who needs a certificate for a particular webinar and you don't hear from us with the certificates, just click on the contact us and let us know. Yeah, so there's a question about whether VectorBase is open to loading more molluscan species. I think this is from Nick Wheeler. So Nick, I think the idea is that just send us an email and let us know about the genomes, and this is something then we will take into consideration along with general priorities and what our program requires, but also in terms of what is important for the community. So if adding more molluscan species is critical and useful for the vector community, then it makes sense that we load those genomes. But it starts by you letting us know about these genomes and why you think they're useful, and that will be the first step. So just click on a contact us link and let us know. All right, I think we've reached the end. We're a little bit over time, and I think lots of people started dropping off. So thank you so much again for joining this webinar. It's been great having all the questions from you. And next week, our webinar will cover regular expressions and motif searches. So if you ever wanted to find a specific motif, either in a protein or in DNA sequence, this will be a very useful webinar for you. We'll teach you about the different tools that you can run in the VEuPathDB resources, how to find these motifs. Thank you so much for joining. Thank you, Eve, for a great presentation. And thanks all for helping, and talk to you soon. Bye-bye.
Thanks, everyone. Bye.
Bye.